Okay.
Welcome everyone to the topic 9 part 2 review for DNA replication. Here's my memes. I hope everyone enjoys them. This one personally is my favorite, that he can only work 5' to 3'. Okay, no one's laughing because they're on mute, but yeah, so let's get into some DNA replication. So Tuesday we left off with the end of replication problem. I did not have slides on it, and I thought we should touch on it because this is definitely a really important concept. So starting with telomeres. So telomeres are specialized structures at the end of the chromosomes. Important to note that not all chromosomes have telomeres. So here's the protective tips over here in this picture you can see. So they're at the end of the DNA. And what is special about these telomeres is that they're repeated nucleotide sequences. So it's over and over, and like for an example, which might help, in humans, the repeated sequence is TTA GGG. And so this is repeated about 3,000 times. So it's not like once or twice, 3,000 times the TTA GGG is repeated. And then telomeric proteins bind to the sequence and loop the DNA to protect it. And so this is the structure we make, it's the T-loop. And this repeated sequence is really important so the proteins know where to bind. And it's also very important for telomerase to know where to bind, which we'll get into next. So yeah, we make this T-loop to protect the DNA because, as you can imagine, if we just have DNA kind of stop here, that's probably not super stable. Oh, sorry. So we loop it to this T-loop structure. And here's some of the proteins. I wouldn't worry too much about necessarily the names of the proteins, just know what they do. And then the chromosome can lose these nucleotides without losing genes, right? So the TTA GGG repeat in humans, like I said, in different organisms, the sequence is different, but obviously we could lose some of these nucleotides because they don't actually code for any genes or anything. They're just there as protective tips. So if we lost some of these nucleotides and some of these sequences, it wouldn't affect, it would affect our genome, but it wouldn't affect how we function because we still have all of our functional genes. But as we talked about in class, these do shorten over time, and we don't necessarily know specifically how it happens or what purpose it serves. But as you age, usually these shorten a lot. So it can become problematic if it shortens too much, obviously. But the question is, how does the cell stop the chromosome from losing too many nucleotides? Like we said, because if we lose too many, then we will start to get into the genetic data that we use to make proteins and such. And then here's my little telomere joke. Not that I think anyone is unfunny, but I liked this meme a lot because it's a repeat. Get it? Okay. So onto telomerase. Telomerase is an enzyme, and it carries its own RNA sequence. And remember that, anyone, what kind of enzyme is telomerase? You can say it or throw it in the chat. It is a reverse I have it on the slide. Cool. It's a reverse transcriptase. And so a reverse transcriptase is essentially what's on the slide here, is the RNA that the telomerase has is used as a template for the DNA polymerase to continue adding nucleotides to the 3' end of the lagging strand. And this picture is really, really good. And so this is to lengthen the template strand. So this picture, the green, is telomerase here. So obviously this is 5' to 3'. Since we're making this 3' to 5', that is our lagging strand. So it binds to the 3' end of the telomere, which in this case, we have our TTA, GGG. So we're going to bind to the 3' end here. And when we bind with this RNA sequence, then new DNA can come and continue synthesizing this direction. And as we continue to do that, we've lengthened over here this telomere, and we've lengthened this sequence. So then the lagging strand can go. Actually, no, because it goes in the opposite direction. So I lied. So we create the new template on the lagging strand, and the leading strand gets synthesized because DNA polymerase is going this way. It's going the opposite way because you can see the arrow, the green. This is the way that telomerase is going versus DNA polymerase is going to, in this case, the left. So we're creating a longer lagging strand, and that's the telomerase is making the telomere. So telomerase enzyme telomere is the repeat sequences. So we're extending that. So then the DNA polymerase can come in and continue matching these to extend the DNA more. And so, yeah, this was the lagging strand that we made more of. And so we complement that and make the complementary pairing. And that's called reverse transcriptase activity because we go against transcription. Does that picture help? Does anyone have any questions specifically on that? I know that that's a lot. So does anyone have questions on specifically what's going on there, the difference between telomeres and telomerase? If not, we have some practice problems we can move on to. Yes, no, maybe so. Okay, feel free, as always, to throw your questions in the chat, public, private, or just shout them out. And if we're good on that and how that kind of protects to make sure we don't lose too much DNA or too many nucleotides, we'll move on to our practice problems with stars. So the first one, we did this one in class, but I want to review it because this was on a problem set from a previous year, which means that this is definitely a really important concept that you should probably know. This is yours, Rosella. You can start with this one. I'm so sorry. Go ahead.
You're so good. Yeah, these problem set questions were written by Laura from last year's problem set. And so did we go over this in class? I didn't know that.
We did it last Tuesday,
Okay.
But it was raining. So a lot of people weren't there.
Okay, so basically you're given this strand of DNA over 3 time points in S phase, right? And you see a different number of origin of replication in each time point. And the question is, how many origins of replication are there in this strand of DNA? So people can just say what they think in the chat, then we can go over it.
And if you've been to my review session before, if you have no idea, you can just private message me or Rosella and say, I have no idea. So that we just know that people are still working on their answers. If you're typing, I don't want to move on.
I definitely struggled with this question in class. And so I had asked her because I was like, which are we looking at a certain one? And so we chatted, and I don't want to give any, I don't if anybody's still working on it, but I finally figured it out and it made a lot more sense. But
yeah, well, if you wanna explain it and answer the question, that would be awesome.
Okay.
See if you get you've got the hang of it, and it will probably help everyone else too.
I think so. So I think more or less you're going to look at the first 2, or that's what we focused on when we were chatting, me and Laura. So in the first one, you see that there's 2 origins of replication. And then in the second one, you see the originals, but then you also see these other 2 that pop up. So in all technicalities, there's 4 origins of replication because in the third time point, all of them kind of merge together. So you see that there's 4 to start with before they kind of merge.
So yeah. Why is there 2 to start and then 4 in the second? How do you know that means there's 4?
Because they don't always fire at the same time. So some of them fire later in a later time point. So that's why you see 2 that pop up later on.
Exactly. Yeah.
Wow, I know what I'm talking about.
Yeah, no, that's perfect. That's a perfect explanation. Yeah, does anyone have any questions on that? Or have anything to add or not understand anything about that? So like Isabella was saying, we look at number 1, and so we can clearly say 2 origins of replication. And then we move on to number 2, because these are time points. We're moving forward in time. We don't move backwards in time. And number 2, we see 2 new ones. So we see this one over here and this one over here, which are new. And that's just because some origins fire later than others. So we end up having 4. And then 3 is later on, and we don't have 3. It looks like 3 here, but it appears that the 3rd one and the 4th one, reading left to right, kind of merged, which ends up happening. That's how we end up having all the DNA copied. If we only had one origin, it would take forever to copy our DNA. We'd be in S phase way longer than we need to be. Does that make sense? Does anyone have any questions about that? Or are we good to move on? Okay, now let's move on to some that I don't think you've seen before.
Okay, so here you're just given a diagram, and there are like different arrows with like numbers, and some of them have like questions in them. So we can just go over them one by one. So question number 1, is this the 5' or 3' end? So
Throw your answers in the chat.
Yeah.
This is an awesome picture. I love this picture.
Mm-hmm. Okay.
Are they going to you, Rosella?
All right. So basically, you know that DNA is made 5' to 3' and read 3' to 5'. So if you're looking at, I just realized you can't see my mouse, but if you're looking at
Tell me what a point.
the last Okazaki fragment, that one. So you know from the arrow that where the 3' and 5' end are going to be, because it's built 5' to 3'. So at the bottom of the arrow it's 5', at the top of the arrow it's 3'. And since the top of the arrow is 3', then you know that the end of that parental strand over there is going to be the 5' end. Does that make sense?
I'm nodding my head as if I was like, yeah. Okay, if we don't have any questions,
right.
Number 3.
3.
So which enzyme? This is number 3 right here.
What is that enzyme?
Beautiful diamond checkered-y enzyme. And Rosella, I can't see what they're saying, so you just let me know.
So this enzyme, yes, exactly. It is a helicase. Somebody answered. Helicases just like separate the 2 DNA strands. And then in eukaryotes, it's the MCM complex that is the helicase. And yeah, that's basically it. It's a helicase. And number 6, what enzyme joins the nick? So remember that there's the nick after the primers are removed in the Okazaki fragments. That remains because the DNA polymerase can't form a phosphodiester bond, since it's like it can't work off of a 5' end. So what is that enzyme that joins the nick? Exactly, yes, ligase. Thank you. So DNA ligase joins the nicks. So for example, Laura likes to ask like some questions. I remember where she asked like, oh, after DNA replication, you have 2 strands, but you see like many nicks in the DNA. What enzyme was non-functional? And then like, DNA ligase. So lots of questions were like, if this enzyme or if this component in DNA replication was removed, what would happen? So that's a good way to study for exams. All right, number 7, leading or lagging strand. So this is kind of confusing. It's pointing to like the whole, it's not pointing to that parental strand. So you see like there are Okazaki fragments over there. So knowing that there are these discontinuous fragments over here, is this the leading or the lagging strand?
Yeah.
Oh, yes. Okay. I got an answer. Yes, it is the lagging strand, right? The leading strand is the continuous one. It goes in the direction of the replication fork. And yeah. Does anybody have any questions about that? Okay. And then number 8, again, which end is this, the 5' or the 3' end? And it's pointing to that leading strand. I'm not getting any answers. But since DNA is replicated 5' to 3', and you know that the leading strand is going to the left, that over there is the 5' end.
Towards the 3' this way.
Yes. Does anybody have any questions about those 1 through 8?
I actually have I think,
Mm-hmm.
I wish I had the speaker notes. No, that's from the next one. Okay, never mind. I have a picture that goes with one of the questions, but it's not this one.
Oh, okay. What kind of nucleic acid is indicated by arrow number 4? And what is the fragment called in the process of DNA replication? So first question, what kind of nucleic acid is indicated?
I'm going to give you guys a hint. It's either DNA or RNA. That's what
a
nucleic acid is.
Nice. Okay. It is RNA. Good. And then what is the fragment of RNA called? Yes, so the RNA fragment is called an RNA primer, and it's added on by the enzyme primase. And this primer is needed because, again, DNA polymerase cannot just start replication de novo from nothing. It needs to add nucleotides to a growing strand. And you're good. This primer is made up of RNA because it is known that the first couple of nucleotides added are error prone. So it's better to have that RNA than DNA, because then it can be easily recognized and removed after DNA replication begins. And yeah, so it's less stable than DNA, so it's a lot easier to remove. And it creates less errors in DNA replication, which is important. Okay, and moving on, what do you call the short DNA fragments indicated by arrow number 5? So I said this word a lot of times.
Say it again. It made it so fun to say if they didn't want to say it all the time.
Yeah.
I'm assuming that's the person that discovered them, or the process.
So the name is Okazaki fragment, right? Just these like short discontinuous fragments. They're discontinuous because they're moving in the opposite direction the replication fork is going. Yeah.
Any questions on this multi-question question?
Mm-hmm. Any questions? No. Okay, everybody got it.
Okay, let's move on. Here's another multi-question question. I can take this one. So true or false? So we'll start with A. When read in the same direction, 5' to 3', the sequence of nucleotides in a newly synthesized DNA strand is the same as in the parental strand used as the template for its synthesis. Is that true or false? Put it in the chat. You can chat to me this time. Last time everyone was chatting to Rosella. Or you can keep chatting to her if you want. I'm not getting any answers. I have one answer. Anyone else want to get an answer in? Well, this person is correct. Yes, so it is false. Why is this false? If we are reading 5' to 3' for both of them, remember the newly synthesized strand and its parental strand end up being DNA in them. So like they end up making a helix. And we know that those strands will run anti-parallel and will be complementary. They're not the same. And if we read them in the same direction, so say we read them like this is a 5' of one strand and this is the 5' of the other, and we read them like this, there's nothing to say they're symmetrical in the first place. So even if we were to read them in the same direction, 5' to 3', there's no reason why they would be the same. And then if we just read them together, they're anti-parallel, so they're complementary. So this is just not true. Any questions on that? I can show a picture for that, and that'll probably make a little more sense. I'll do that when we go to C. So for B, each time the genome is replicated, half of the newly synthesized DNA is stitched together from Okazaki fragments. Is that true or false? What do we think? Any ideas? If you don't know, say don't know. I don't want to cut anyone off if they're thinking. No one's saying anything. There's one. So we got one true. Does anyone agree, disagree? True? Do we think it's true? Well, everyone's being shy tonight, but yeah, so this is true because when you think about it, half of the strands that you're synthesizing, for every continuous strand you make, you're going to have a lagging strand. So you will also be making Okazaki fragments half of the time. And then we ligase those together, or in this, stitch ligase, we connect them. So, and that happens half the time, and half the time it's continuous. So if you think of it that way, every time our genome is replicated, half of the new DNA was made by the Okazaki fragments because half of the time we have a lagging strand. If that makes sense. For C, when bidirectional replication forks from adjacent origins meet, a leading strand always runs into a lagging strand. Is this true or false? This is kind of a loaded one. You might need to draw it out or think about it. I totally didn't mean to go to that picture yet. So looking at replication forks from adjacent origins, so origins next to each other, does the leading strand always run into the lagging strand? I got one answer. Anyone else? Yeah, so true. So this is the picture I've been waiting to get to. So if we look at this, and we look at, say, this replication fork by itself, and we're going to the right, it's going to be the leading strand. And if we're going to the left, we're going to look at the lagging strand. It's going to be the lagging strand. Now, for the entire DNA, if we have another origin over here, going to the right for the green side of the DNA, going to the right will always be the leading strand, and going to the left will always be the lagging strand, because for the entirety of the DNA, it's 3 to 5. Like the directionality of this side of the DNA never changes. So if you think about, if we had a replication fork here, going to the right, we have a leading strand, and going to the left, we always have a lagging strand. So when they meet, I don't know how computers work. So when they meet in the middle, the leading strand will meet the lagging strand. And the same for this side. We have our lagging strand here. And if we had another origin here, we know that to the right, there's always a leading strand. So when they run into each other, the leading and lagging strands always run into each other. And then I was going to look at this picture for A2 just to show, if we just look at this piece of DNA, ignore the replication, which is hard in this picture, but if we're just looking at 2 strands, when it says reading 5 to 3, if we read, we're looking at the brown strands right now, 5 to 3 this direction, and 5 to 3 on this strand, there's no reason they would be the same, because that's assuming that there's some like middle point and they're symmetrical. So it could be T, A, G. Nothing indicates that this would be T, A, G reading this way. Now, if you were reading them like this, 3 to 5 and 5 to 3 for complementary pieces, then it'd be complementary base pairing. But if you read them 5 to 3, both of them, there's no rhyme or reason necessarily for what it would look like, unless maybe it's like a telomere, but even then it's a little tricky. I think that kind of helps to visualize A. So for A or C, does anyone have any questions before we move on to D? I know that this directionality stuff is kind of tricky. It's one of the harder parts of this unit. Everyone good on those? Okay, so for D, if an origin of replication is deleted from a eukaryotic chromosome, the DNA on either side will ultimately be lost as well, because it cannot be replicated. Is this true or false? Got one answer. Anyone else? Any thoughts? Well, the one answer I got was false, and that's true. If we look at this picture, wrong way, this picture right here, if you notice, there were 4 origins here, and we look here and there's only 3 pockets. And that's because the origins are going to run into each other. So say that we lose this origin of replication right here, there's nothing to stop this origin and this origin to end up meeting and connecting the DNA. And that's just how DNA synthesis works, is we have multiple origins that end up running into each other. Like in part C, replication forks are going to meet up. So it might take a little longer if we lose an origin. There's a reason we have so many origins, and that's for speed and efficiency. But just because one origin of replication is deleted does not mean that the genes on either side will be lost. They'll still be replicated. It just might take a little longer. And I can't give you any time frame of what a little would be, because this is all happening super fast. So yeah, do we have any questions on these true falses? Everyone have the answers down. We're good on that? As always, feel free to put it in the chat or stop us. And if we have any questions, we can come back. And the next one, that's the picture.
Okay, so, whoa, why was there an echo? Okay, so DNA polymerase processivity, processivity just basically how well the DNA polymerase can attach to the DNA and like replicate before it falls off. And we learned that the sliding loader and sliding clamp help to make the DNA polymerase more processive. So the question is saying, you wish to carry out an experiment on a mutated bacterial polymerase. 3, the main replicative DNA polymerase in bacteria. The mutation is in a region of the protein that you suspect weakens the binding of the polymerase to the sliding clamp. Devise a hypothesis for how this mutation might affect DNA replication and explain your reasoning. So this might be hard to answer on the chat, so I can just talk about it. But basically, if there's a mutation that weakens the binding of the polymerase to the sliding clamp, that means that the DNA is going to be less processive, which means that it's going to fall off the DNA more often, right? And if it falls off the DNA more often, the sliding clamp needs to bind to the DNA again, and the loader needs to put it on and everything, so which takes time. So the replication is going to take longer, it's going to be less efficient, which is basically what's going to happen. The DNA can still be replicated, but it's just going to take longer and it's less efficient because the DNA polymerase keeps falling off since there is a weaker bond between the polymerase and the clamp. So that's basically it.
This is a great question to study to make sure you understand the process of how you get there, because this looks a lot like exam questions. As you've seen, we love to throw experiments in there, and if you notice something or if you change something, what's going to happen? So this is probably one of the closer ones to what you're going to see on an exam, but they're all problem set questions from the past, so they're all equal. Any questions on this? This is our last question we have, but I did have a question in the forum that I want to go through. But do we have any questions on this before I have to switch slides? So slideshows. As always, feel free to stop us, but I'm going to switch to, I had a question about bonds and the different bonds that are made between different things. So I know that I don't know exactly the problem set question, but I do know that there was some things about bonding. So we're just going to go through quickly all the bonds that are involved in DNA and DNA replication. So I'm trying to find, where's my favorite? Let's do this one. This one. This one. This is a less blurry version. So let's do this one. So I'm not going through, these are the slides that I went through on Tuesday. Feel free to watch that video on YouTube. And so starting with the bonds between the different things in each nucleotide. So we have a phosphate connected to the sugar here. So this is right here. Well, this and the base, this is a nucleotide. So there is a bond created between the sugar of one nucleotide and the phosphate of the other, and that bond is right here. And that's our phosphodiester bond. That's the bond of our backbone. And these phosphates bond to the sugar of a different nucleotide. So these are covalent bonds. And, oh, sorry. Brazil, I just got so confused. So this is the backbone bonds. So we make these, and I'm going to go a little more in depth into how, when we're replicating the DNA, obviously, because we're adding nucleotides. So we're going to make that backbone. These are covalent and these don't break, which makes sense because the backbone, whether we unwind the helix or not, the backbone's not going to change. And then we have our hydrogen bonds between our actual bases. So between adenine and thymine, we have 2 hydrogen bonds, and between cysteine or cytosine, not cysteine, cytosine and guanine, we have 3 hydrogen bonds. This is important. I don't know if this is going to be an exam question, but I swear I've seen it before. Why do we have the Tata box as the start of replication is because it requires less energy to break these 2 bonds than it requires to break these 3 bonds. And so the thing about the hydrogen bonds is that we know they're non-covalent, so we can break them. Covalent bonds, it takes a lot to break. Or non-covalent bonds, we can break, but in numbers, they're strong. So when it comes to DNA, we want a strong enough bond that we're going to be able to hold everything together, but we also want to be able to break it so that when we need to replicate, we can unwind and break it. So hydrogen bonds during DNA replication, we break them and we make them, right? Because we break them when we open up to replicate, and then we make them when we add a new nucleotide. And so on to the phosphodiester bonds. Specifically, what is happening? Oh my gosh, my computer's freaking out. So we have 3, I'm not clicking anything. Does anyone know why on earth that's happening? So we have 3 phosphates here. And the way that the phosphodiester bond is made is that the oxygen, and you don't need to know this chemistry necessarily, but my mind works in chemistry. This oxygen attacks the phosphate that's attached to the sugar, which then releases 2 of the phosphates. So we're breaking a bond between the 2 phosphates here, and they're cleaved and they're sent away. We don't have to worry about what those are doing necessarily, which then makes our phosphodiester bond. So we're making a bond between our base and our phosphate, but we're removing the bond between the 2 phosphates. And this backbone, we don't break. I guess unless we have to degrade that. We haven't talked about a situation in which we have to break the backbone. So breaking this is breaking a covalent bond. This is making a covalent bond. And then we have our hydrogen bonds between our bases. And those are the bonds that are relevant in DNA replication. Are there any bonds, anything else that I'm missing, or any questions anyone has relating to that?
I have
yeah.
a question. Okay, so the bonds between the phosphates are broken, which I think I understand because they disappear, or not disappear, but they break apart and then they're
and then those 2 phosphates, you don't have to worry about what they're doing.
You don't have to work. Okay, so
they go away. They go away.
The bond gets broken.
Yes.
So then the bond between the phosphate and the bases.
Sugar. Have I been saying base? I'm so sorry. This is the sugar.
No,
the base.
No, I'm looking at
base.
Technically, I'm looking at our problem set, and I'm just looking at the different combos she has here.
I just went through the ones that are relevant, and anything else, I don't think I'm forgetting any. I will say that, because I know that there's an option D, and that's that there is no breaking or making of bonds. So we have the sugar and the phosphate, we have the phosphates, and then we have the hydrogen bonds between the bases.
Hydrogen bonds between the bases, yes.
Because this is the base, and so does this base ever interact with the phosphate? The base only interacts with other bases. I mean, the base is connected to the sugar, but the nucleotide never breaks apart. The phosphate, the sugar, and the base will always stay together for nucleotide, but the base, the only thing the base really interacts with is other bases. Okay. And the phosphate attaches to the sugars and the phosphates interact. And then when it's talking about any phosphate interactions, that's regarding these phosphates. Obviously, the phosphates that are on the backbone aren't interacting because it's sugar, phosphate, sugar, phosphate.
Okay.
Anything else?
I don't think so.
Does that make sense?
Yes.
Knowing those interactions and then knowing why there's not other interactions.
I'm still fuzzy on all of them. They're just it's a lot to think about. So I am still struggling with that, but it might just be I have to work with it.
It also just takes time to there's a lot. We're doing a lot tonight. So I always recommend writing it down and then looking at it the next day. Once your brain goes fuzzy and you're just like, I can't think anymore, you just put it down and you come back to it the next day and study a little bit every day.
Yeah.
So does that help answer the bonds question? Anyone have any other questions about bonds? James Bond. As was the answer to the iClicker question. Yeah, so any questions on the bond or anything else that we want to go through before we stop recording? That can be anything from Tuesday, the slides from Tuesday, anything from today, anything in general. That would be for the good of the group. Because if not, I think that we can then stop the recording, Rosanna. Bye everyone on YouTube. Miss you dearly.